couple of reminders and announcements. One is that I've been asked to remind you that applications for summer research on campus are due tomorrow. And you go to Handshake. If you have any questions about the Handshake, I actually have the Handshake link somewhere on my computer. I can give it to you, or you can get it from Peter Blair or from Sarah Paul. One of those people will be happy to share with you the Handshake link for applications to summer research. Reminder that Quiz 1, A&P Quiz 1 is due tomorrow. Pick it up at the CST desk. Due at the CST desk by 11:30 tomorrow night. I will post the answers, my answers to the lab worksheets from the first lab are up on Moodle now. And my answers to the second lab, which is happening today, the last of the second labs are happening today, I'll post that at the end of lab today. So my answers will be available on Moodle, but you have your answers from your lab, or at least you will, all of you will by the end of the lab period today. Quiz 1 due tomorrow at 11:30 p.m. CST desk. Quiz 2 already will be available on Friday and is due next Wednesday, so a week, a little over a week from now. But it's due at 4:30 p.m. because this is the day before early semester break starts on Thursday, and the library, the desk is closing at 5 p.m. So you have to pick up and turn in your quiz before the desk closes at 5 p.m. So that's why I'm setting a 4:30 p.m. deadline next Wednesday. Not tomorrow, but next Wednesday. Today is what? I don't have my calendar in front of me. Do we want to look at a calendar? Yeah, that's Thursday. Early semester break is really early. Why? Yeah. You have a question?
Yeah, how much is the quiz worth? Is it like half the thing?
On the course value scale, homework, problem sets and quizzes are worth 50 points each, and an exam is worth 125 points. That, out of 1,000 total for the course. Okay. Any more questions? So today we're going to talk about some specific neurotransmitters and specific neurotransmitter receptors. So we're going to take what you know from a general perspective about neurotransmitter and neurotransmitter receptors and start to apply them to some specifics. We'll learn about some neuroanatomy related to dopamine, norepinephrine, and serotonin neurotransmission, and we'll introduce some terminology of neuroanatomy. It's hard. So here is a table which is available to you on Moodle on now 2 of your handouts. I don't really want to go through it in great detail because it's available here. Let me just pick up the main points. And maybe circle the points that I think you should know. The words and the terms that you should know. So, I mean, you should know the names of the neurotransmitters. I'm not going to circle them. You should know the 2 types of acetylcholine receptors and their names. There is a nicotinic receptor, which is a ligand-gated ion channel, or an ionotropic receptor. So an ionotropic receptor is a neurotransmitter activated channel. And you should know that there are GPCRs for acetylcholine also. So acetylcholine can activate ionotropic receptors or they can activate GPCRs. The ionotropic receptors are called nicotinic receptors because nicotine is actually a good agonist of these receptors in the peripheral nervous system. It's actually a pretty good agonist of nicotinic receptors in the central nervous system as well. Okay? And we can get into why nicotine is addictive because of its ability to activate nicotinic receptors in the brain. The GPCRs are called muscarinic because muscarine is a toxin from mushrooms that blocks these receptors. So the names are pharmacological. But I will use them, so you should know them. And you should know that acetylcholine is excitatory at the neuromuscular junction, at the synapse between motor neurons and muscles. But you should know that it can be excitatory or inhibitory in the nervous system. Okay? Dopamine and norepinephrine dopamine, norepinephrine, and serotonin are all monoamines. That's the class of chemical that they are. And dopamine and norepinephrine are catecholamines. Catecholamines are a derivative of tyrosine, but I'm not certain on that because I'm not going to test you on that. But I will expect you to know that dopamine and norepinephrine are catecholamines, but that serotonin is another monoamine that is not a catecholamine. It's not a tyrosine group. I will want you to know that the cell bodies of major dopaminergic neuron pathways are in the substantia nigra and the ventral tegmental area. Those are names of parts of the brain. This is getting into neuroanatomy. Parts of the brain where dopaminergic neuron cell bodies live. The cell bodies of major neurons that use dopamine as their neurotransmitter are located in these two parts of the brain. And we'll see them on some neuroanatomy slides. I do want you to know that the location of cell bodies of norepinephrine releasing neurons is in the locus ceruleus. It's a name, a neuroanatomy name that I want you to know. And the same thing for the raphe nucleus for serotonin releasing neurons in the brain. That's where the cell bodies are. That's not where all the synapses are. It's where the cell bodies are. I do want you to know that dopamine activates two different types of GPCRs, GS and GI. But I don't care that you know which one is which. Okay? You don't have to memorize that D1 receptors are GS and D2 receptors are GI. You don't have to memorize that. But I do want you to know that there are two classes of GPCRs that are activated by dopamine. They're both GPCRs. You know all about them. And one of them is coupled to GS, and the other is coupled to GI. Same sort of thing for norepinephrine. They are coupled. They activate GPCRs, not ionotropic receptors, not ionotropic receptors, not ionotropic receptors. And I do want you to know that all three of the major types of GPCRs that we have talked about can be activated by norepinephrine, but I don't care that you know which one is the alpha adrenergic receptor and which one is the beta adrenergic receptor. And I do want you to know that serotonin can activate many different types of GPCRs and one type of ionotropic receptor. We will talk about some of the effects of dopamine with regard to motor control, reward and addiction, and Parkinson's. We will talk about them, but I'm not going to test you on knowing that dopamine is responsible for Parkinson's. If I ask you a question about Parkinson's, I'll give you enough information to know we're talking about dopamine. And we'll talk about how norepinephrine is important for general arousal, and how serotonin is important for mood and depression. Okay, so the table has more information, so it can be used as a reference, but I think I've given you indications of what bits of information you want to know for the exam. Again, the quiz 2 will be anatomy, and I'm not calling this anatomy. Alex?
For acetylcholine, should we know the GQ and GI, or is that?
Well, so I guess to be symmetrical, no, I don't care. I didn't circle it, so I don't care. No, I do care. I'm not gonna, I care a lot, but I'm not going to make you care. I'm not going to make you memorize it. Okay, so we can do the same sort of thing for some other major neurotransmitters. So let's go through this again, maybe a little faster, just because you sort of know the drill now. I do want you to know that glutamate, an amino acid neurotransmitter, I want you to know that glutamate is the major excitatory neurotransmitter in the nervous system, that it activates two types of ionotropic receptors called NMDA receptors. They're called NMDA receptors and AMPA receptors, but I'm not going to ask you to memorize the names, but that there are ionotropic receptors to glutamate, and there are GPCR receptors to glutamate. I do want you to know that GABA is the major inhibitory neurotransmitter in the nervous system. We have excitatory and inhibitory neurotransmission, EPSPs and IPSPs, and GABA is the most important inhibitory neurotransmitter. And it also activates ionotropic and GPCR receptors. Glycine is the amino acid neurotransmitter in the spinal cord that acts just like GABA. So I'm going to circle glycine, and I'm going to circle spinal cord. It's ionotropic only, and it's inhibitory. Now we're going to get into some other neurotransmitters that are, important, probably the most important thing to some people, but I'm not going to circle anything. So ATP and adenosine are, believe it or not, are neurotransmitters. They're the purine neurotransmitters. And they activate receptors, and they can be either excitatory or inhibitory, but you see I'm not circling anything. This is now information on your slide for reference, but not for memorization. So are there questions about these classical neurotransmitters? The ones from the previous slide and this one about the terminology and the physiology. Physiology is the type of receptor, terminology is the words. Terminology and the physiology of the classical neurotransmitters. 4 on this slide, 4 on this slide, but only 3 of which you have to devote any effort to. So now let's talk about receptors. And there's a bunch of information. And I'll circle the things that I think are most important. Acetylcholine is synthesized as a combination of choline. So this is choline. I'm sorry, I circled it, but I don't expect it to know it. This is choline, and this is acetyl. So acetylcholine. And unlike all the other neurotransmitters that we're going to talk about, which have uptake mechanisms like the dopamine uptake mechanism and the serotonin uptake mechanism and the norepinephrine uptake mechanism, acetylcholine is hydrolyzed in synaptic clefts by acetylcholine esterase. So acetylcholine is broken down in the synapse, not just reuptaken. Choline is then reuptake by a sodium choline cotransporter, just like the sodium norepinephrine cotransporter at norepinephrine synapses, or the sodium dopamine cotransporter. But in this case, we're taking up not the neurotransmitter acetylcholine, but its bit, choline. Cells have a ton of acetyl, right? Acetyl-CoA is the way acetyl is in cells. And so what's reuptake is choline, and then acetylcholine is resynthesized, and then repackaged into neurotransmitter vesicles. Okay, so that's some information about acetylcholine. Here is the molecular structure of the nicotinic acetylcholine receptor. How do we know it's nicotinic and not GPCR? It doesn't look anything like a GPCR. It's got a lot more than 7 transmembrane domains. And its side view and its top view shows that it's a pore. Transmembrane, intracellular, extracellular domains. Where acetylcholine binds is in the extracellular domain, and it's actually in these pockets right here. I'm not circling it because I'm not expecting you to know, but just that is the knowledge. I'm applying some of your cell physiology knowledge about protein structure to an important molecule, the nicotinic acetylcholine receptors, ionotropic receptors. So ionotropic receptors are the nicotinic ones. Metabotropic receptors is another name for GPCRs, which I will use. So when I say ionotropic, you know it's a ligand-gated ion channel. And when I say metabotropic, you know it's a GPCR. Just another name for a GPCR, metabotropic receptor. And like I said, there was the muscarinic kind. So here is a cartoon of a muscarinic receptor. And so if you see this bundle of 1, 2, 3, 4, 5, 6, 7 helices, you can be pretty darn sure that it's a GPCR, metabotropic receptor. And like I said, there are metabotropic receptors for acetylcholine that are coupled to GQ and to GI, and you know the stories. But you don't have to memorize, you do not have to memorize that M1, M3, and M5 metabotropic acetylcholine receptors are coupled to GQ, and M2 and M4 metabotropic acetylcholine receptors are coupled to GI. Don't do that, but here it is on the slide. If you want to look it up and just say, oh, you've just read a paper about the M3 muscarinic receptors, which one was that? You can go to the slide and say, oh, M3 is coupled to GQ. And then you know, okay, GQ, there's going to be some change in calcium. There's going to maybe PKC is going to be activated. Take your knowledge of the GQ receptors, which you know now, or will, and apply it to muscarinic GPCR. So now, okay, I'm going to transition to using the language of monoamines. So we have two kinds of monoamine neurotransmitters, the catecholamines and the non-catechol monoamines. And the most important non-catechol monoamines is serotonin, which is also known as 5-hydroxytryptamine or 5-HT. So if you see 5-HT, you're talking serotonin. They're just synonyms. This is the catechol. Not circling it. This is the catechol moiety. This dopamine, this tyrosine derivative is what makes this a catecholamine. So this is the structure of dopamine. Dopamine has two critical pathways in the brain that I do want you to know. I'm getting ready to circle something. There's a pathway from the substantia nigra to the striatum. We'll talk about that from a and here's the names. We'll see it on a neuroanatomy slide in a few minutes. Substantia nigra to striatum is a major dopaminergic tract or pathway. The cell bodies and the cell bodies are in the substantia nigra, and the synapses, where it's released, is in the striatum. This is where the cell bodies are. This is where the synapses are. Or at least some of the synapses. There's another one from the ventral tegmental area, or VTA, to the nucleus accumbens. This one is critical for motor control and is defective in Parkinson's disease. This one is important for reward pathways, feel-good pathways, and so they are important for addiction, which is an abnormal activation of reward pathways. So, motor control and reward. We'll talk about schizophrenia. Maybe if there's time, we'll talk about schizophrenia and Parkinson's as abnormalities in dopaminergic signal. So here is a cartoon of the brain. What's the name of this section? It's a medial sagittal section, cutting the brain down the middle, looking at it from the side. The section is a sagittal section, midline sagittal section. And we'll start to use some of the language of neuroanatomy now. So here's the neocortex, frontal lobe, parietal lobe. I'm just going to use it. We'll see it on, and you'll see some words circled on another slide. Parietal lobe, occipital lobe, cerebellum. This is the brain stem, which has 3 parts: midbrain, pons, medulla. But for dopamine, the substantia nigra and the ventral tegmental area are in the midbrain. They project widely, so those dopaminergic neurons go lots of places, right? So this is where the cell bodies are. These arrows represent axons. The arrowheads represent where the synapses are, where the neurotransmitter is released. And the one we're talking about is, the 2 that we're talking about is from the substantia nigra to the striatum. And the substantia nigra to the nucleus accumbens, which is not actually shown on here, but it's somewhere over here. Questions about dopamine? Dopamine is a catecholamine. So here's the here's tyrosine. Here's catechol. Here's the catecholamine. The same pathway is used to synthesize dopamine, norepinephrine, and in the peripheral nervous system, epinephrine. So dopamine and norepinephrine are the 2 catecholamines that we're talking about here, and they're all part of the same pathway. I'm not asking you to memorize the synthetic pathway for dopamine or norepinephrine, but what's important on this slide is that they are both tyrosine derivatives. They're derivatives of the amino acid tyrosine. And dopamine is a precursor to norepinephrine. Dopamine is a neurotransmitter in its own right, but it's also a precursor to another neurotransmitter, norepinephrine. They're both catecholamines. Not a new language. Let's talk about dopamine receptors. They're all metabotropic. They're all GPCRs. So they all look like this. Some of them are coupled to GS, and some of them are coupled to GI. But you don't have to know which is which. But you might, just for future reference, you might know that if somebody is referring to a D1 receptor, it's a dopamine receptor. It's just the language of dopamine receptors. D1Rs, D3Rs are dopamine receptors. And here's a slide that will help you unpack. If you read about a D3 receptor, you'll say, oh, that's coupled to GI. You can just put it. So dopamine receptors are located widely in the brain, but they're also, but let's talk about one specific location where dopamine receptors are located, in the nucleus accumbens. And nucleus accumbens is part of that reward pathway. And some rodents have a property of monogamy. And they have that property of monogamy in part because their nucleus accumbens gets more dopamine receptors. I mean, this is a funny slide with these parts, but more dopamine receptors in the nucleus accumbens of the paired animals than in the unpaired animals. And this increase in expression of dopamine receptors is part of the pathway that leads to reward. This is a rewarding experience for these voles. And I mean, I'm not going to say that monogamy is an addiction, but it's a rewarding experience for bulls. Abnormal activation of the nucleus accumbens by other rewarding pathways, whether we're talking about gambling or drugs, can also activate the expression of dopamine receptors in the nucleus accumbens, and that's part of the sort of extreme manifestation of a reward pathway, which is called addiction. I don't know why I put this one again, so let's go on. So now we're moving on to norepinephrine. Just want to make sure I can space. Yeah. Okay. Now we're moving on to norepinephrine, another monoamine catecholamine neurotransmitter. The most important location of the cell bodies of norepinephrine releasing neurons, noradrenergic neurons, is the locus ceruleus. So the jargon here gets complicated. Norepinephrine is the American name for the neurotransmitter. Noradrenaline is the British name for the same neurotransmitter. And the Brits got to name the receptor, so they're called noradrenergic receptors. Adrenergic receptors comes from the name adrenaline, which is the English, British name for what we call norepinephrine. So there's no such thing as a norepinephrinergic neuron. We just don't call it. It's a noradrenergic neuron. Anyway, the cell bodies that contain, of the cells that are noradrenergic, one of the important locations is the locus ceruleus, which is also in the midbrain. Let me go back. So, substantia nigra, does anyone know enough Latin to know what that word means? What those words? Substantia means substance. Nigra means black. Black substance. There's a location in the brain that's dark, black, and colored, and so that location is called the substantia nigra. Locus ceruleus. I'm not going to test you on this jargon, but just it's sort of interesting. Locus means location, a place. Ceruleus means blue. So this is the blue place. So there's the black stuff or black substance and the blue's place in the brain. The locus ceruleus is the locate, and I've never seen it with blue, but maybe in some maybe in a fresh, unembalmed brain, it might look bluish in color. I really don't know. Anyway, the locus ceruleus, the name, in the midbrain is the location of the cell bodies of the noradrenergic neurons. Axons from the locus ceruleus go everywhere. And that's why noradrenaline is an important neurotransmitter of general arousal. I'm not talking specifically about sexual arousal or anything. I'm just talking about being awake and alert arousal. Adrenergic receptors, the receptors for norepinephrine, are metabotropic. They are GPCRs. They look like this. There are several different subtypes of adrenergic receptors, alpha and beta. They're subdivided even further, alpha 1 and alpha 2, beta 1, beta 2, beta 3. You can see I'm not circling anything. They are coupled to GQ, GI, and GS, but you don't need to memorize which is which. So I could ask you a question on an exam saying, you are studying the alpha-2 receptor in a set of neurons. As you know, the alpha-2 receptor is coupled to GI. And then give you some problem to solve with that knowledge. I would tell you that we're talking about GI. Okay? But then you have to apply your knowledge of GI to maybe solve a problem related to GI coupled receptors. Or I could talk about alpha-1 and say, as you know, alpha-1 receptors are coupled to GQ, and then ask you to solve some problem knowing what GQ receptors do. Okay. Serotonin is a monoamine. Where's the amine? One of them, monoamine, but it's not a catecholamine. It's not a derivative of tyrosine, it's a derivative of tryptophan. The cell bodies, a location for a lot of cell bodies that, of the neurons that release 5-HT as their neurotransmitter, is in the Raphe nuclei, again, of the midbrain. And they go everywhere. And so they're important for a number of things, especially related to mood. 5-HT receptors are, well, some of them are metabotropic. Some of them are ionotropic. I'm not asking you to know which is which. So I will not ask you to memorize that 5-HT3 receptors are ionotropic. I won't do that. But I might say 5-HT3 receptors, the ionotropic serotonin receptors, are sodium permeable. And then ask you to solve a problem related to what activation of a sodium permeable ionotropic receptor might do to a neuron. So the GPCRs are coupled to all three of the, different receptors. 5-HT2 are coupled to, I've forgotten, so I don't even know. 5-HT2, 5-HT7, 5-HT9 are coupled to different G proteins. And so they do different things. And then, so remember, I'm just making this a little interesting to you, is that the SSRI class of antidepressant drugs, markers are always a little mark on the screen, the SSRI class of drugs, of antidepressant drugs, acts to inhibit the serotonin reuptake transporter. The serotonin reuptake transporter reuptakes serotonin, and then a vesicular monoamine transporter repackages the serotonin into vesicles. This is a proton exchanger. This is the proton pump. This is a neurotransmitter proton antiporter. And this is a mistake. This is a serotonin sodium co-transporter. I should fix this on the slide. And so what kind of receptor is this cartooning? Would we cartoon a metabotropic receptor this way? No. This is the cartoon of the ionotropic receptor, or the 5-HT3 receptors. Interestingly, serotonin, so 5-HT3 receptors, are pretty similar in molecular structure to some of the nicotinic acetylcholine receptors, which are also ionotropic. And so drugs that are sometimes used to treat nicotine addiction, like Chantix, is a drug that is targeting the nicotinic receptor, but it has side effects on the serotonin receptor because of the similarities of the two receptors. And that's why Chantix which is the drug used to try to help people avoid the cravings from nicotine, can cause nausea because it activates serotonin receptors that are associated with movement and motion sickness. I'm not circling anything, you don't have to know that, I'm just trying to make it interesting. So, in addition to the classical neurotransmitters, so let's see, what classical neurotransmitters have we talked about? Acetylcholine, dopamine, norepinephrine, serotonin. We have not yet talked about glutamate and GABA, but we will. But I want to skip to a class of neurotransmitters called the peptide neurotransmitters. The peptide, so some neurotransmitters are not small molecules like amino acids, glycine or glutamate, or GABA, and they're not small molecules like norepinephrine, catecholamine, small molecules like norepinephrine and dopamine. They're small peptides, and they have an amino acid sequence. And you may have heard about endorphins and enkephalins. So these are peptides that activate peptide neurotransmitters. Activate peptide neurotransmitter receptors. And almost all of them are GPCRs. I will never ask you to know, the sequence of any of these, right? I'm not. But I do want you to know that there are peptide neurotransmitters. They're sometimes called neuromodulators because they sometimes are released along with the classical neurotransmitters. So I guess we're going to come back to glutamate and GABA. But you already know the concepts from early on. Glutamate activates both ionotropic and metabotropic receptors. GABA activates both metabotropic and ionotropic receptors. Glutamate, so knowing that glutamate is an excitatory neurotransmitter, and knowing that it activates ionotropic receptors, what ions do you think go through ionotropic glutamate receptors? Sodium. And you know that from, well, you don't know that from the problem set, but you could have guessed it because that was the nicotinic acetylcholine receptor. It was the sodium permeable excitatory neurotransmitter receptor. GABA-A receptors activate chloride ionotropic receptors, and that's what makes them inhibitory. We did that in the problem set. GABA-B receptors are the metabotropic ones. They activate GI, which activates a potassium channel. And that's what makes them inhibitory. All right, let's take a break now because we're going to transition to neuroanatomy using the language of neuroanatomy. So let's take a short break now. I don't teach a lot of neuroanatomy in this room. Most of the a lot of anatomy. Most of the anatomy you learn is in the A&P lab, and from doing the coloring book, the homework, and the A&P revealed. But it's useful to use the language in this room as well, so that's what I'm going to do for the rest of this class today. So here, this is a cartoon from your textbook. This is a lateral view of the brain. Right? So this is looking at the brain from the outside, from the side, lateral view. And this is a midline sagittal section. Just about everything on this slide represents language that I do want you to know. So the only thing only language that I don't use is, I do use it, but I won't, well, cerebrum we certainly will use, but diencephalon, I don't use forebrain very much, so let's cross that. I don't care that you memorize the word diencephalon. It's the cerebral cortex. So the lobes of the cerebral cortex are the frontal lobe, parietal lobe, occipital lobe, and temporal lobe. This is all words to know. The brain stem is comprised of the midbrain, where the substantia nigra is, the locus ceruleus is, the raphe nuclei are. Those are all in the midbrain. Below the midbrain in the brainstem is the pons, and below the pons in the brainstem is the medulla oblongata. Connected to the medulla is the spinal cord. Cerebellum, here's the cerebellum, here's a midline sagittal section of the cerebellum. Okay, so these are all words to know. It's part of the language of neuroanatomy. And so, on a quiz, not quiz 1, I'll be clear, but on quiz 2, I could ask you to identify structures on the brain, either from a lateral perspective, or a sagittal section, or in a few slides you'll see coronal sections. So, and here is a top view. So let's start to use some of the language of neuroanatomy. These lumps bulges of cortical tissue, of cerebral cortex, one of them is called a gyrus. That's the singular, and the plural is gyri. One gyrus, two gyri. The grooves between gyri have a name. One sulcus, two sulci. Okay? So sulci separate gyri. Some of them have specific names. So this is anterior. And this is posterior in this view. And I will always, because sometimes it's hard to tell, I'll make sure that you know which is the front and which is the back, which is the top and which is the bottom. That's easy. Front and back sometimes is good. This thing, this structure, this sulcus that separates the two cerebral hemispheres has a name. Does anyone know what the name of that one is? It's called the longitudinal fissure. Okay, it's got a name. What is the shadow? Keeps walking. Let's see what it's got. What is that I just tried to outline right there? What's its general name? A sulcus. It has this one has a specific name. It's called the central sulcus. And if I were to ask you, can point to the central sulcus, and you were to point to this one, I would say that's close. Yes?
I don't have a slide in notes.
Okay. The central sulcus, so the gyrus in front of the central sulcus is called the precentral gyrus. So even if you don't have this picture in your slides, you could be writing down these names and looking them up. And the sulcus, the gyrus behind the central sulcus is called the post-central gyrus. Okay? That one has a specific name. Okay, so what's the name of this lobe? Frontal lobe. And what's the frontal lobe? Is separated from the occipital lobe by the central sulcus. So the precentral gyrus is the most posterior gyrus of the frontal lobe. And the post-central gyrus is the most anterior gyrus of the parietal lobe. So this is frontal, this is parietal. Back here, we're going to see occipital. We can't see temporal on this picture. So let's, okay, so let's take what I just took from this slide that you don't have and apply it to this slide, which you do. And I know you can't read the tiny print here, but because that's not, I mean, here's the answer key to these names here, and it's actually in Latin rather than in English. So it's, but it's, so if you want to know the Latin words for these things, you've got it on the Moodle. But here are the words that I do want you to know. So we've talked about a sulcus, and the sulcus in general, and the central sulcus in particular. Sulcuses are everywhere. Sulci are everywhere. Central is a specific one. We've talked about a gyrus, and then we've talked about two specific gyri. We've talked about the longitudinal fissure. Now we're going to get into some new ones. So I have a laser pointer, and I want to offer it to somebody who wants to show me the central gyrus, the central sulcus, on the image that you have in front of you, not on the image that is in the slide before. Anybody willing to take this laser and point to the central sulcus? Yeah, let's do this together. I think that's a pretty good bet. So that now I'll know. No, because, well, so it is saying that, so I would have accepted it because I think that was a good choice, but it is this one is in fact the central sulcus, because I looked up D and it says D is the pre-central gyrus. Oh, and I didn't, so this is obviously the front. No, let me use the anatomical words. This is the anterior and this is the posterior, right? So this is the central sulcus. This is the pre-central gyrus. What's this? Post-central gyrus. Okay? Can we see the longitudinal fissure? Not really. We certainly can't see it here. And the longitudinal fissure is where this cut was made. So if I had to say, if I had to point to the longitudinal fissure on these pair of images, I would have to say, well, it's here. You know, I wouldn't try to make you do that in an exam setting, because how would you point to it if that's all you're seeing? Okay, so now let's talk about some of the other terms to know. The lateral fissure. Does anyone So I haven't taught this one yet, but you can make a guess. Maybe even trying to read the Latin. The lateral fissure. Oh, you hit it there. No, not that one. Lateral. That one. That's the lateral fissure. The fissure that separates the temporal lobe from the frontal and parietal lobes. So lateral is on the side. And we only see it on the side. If I go back to the previous slide, we can't see the lateral fissure in this picture, but we can see it. There's the lateral fissure. Okay? Is it a gyrus or a sulcus? The lateral fissure. It's a sulcus. So some people call it the lateral sulcus. Yeah. The lateral fissure separates the temporal lobe from the frontal and the parietal lobes. Okay, so let's see. Terms to know. We've covered sulcus, gyrus, longitudinal, lateral. Lobes, we've talked about the lobes. Frontal. So which ones can we see here? What's the name? So somebody point to the frontal lobe on both pictures. And we can do it over here too. We can get this side of the room full. There's the frontal lobe in the midline sagittal section. And where's the frontal lobe there? There's the frontal lobe there. Okay, somebody point to the temporal lobe in both pictures if you can, or just say, no, I can only see it in one picture if you can't. Anybody? Yes, sir? Right, temporal lobe. Ah, so we are, you're right. This is the temporal lobe from the side. This is we're looking at the back temporal lobe. We're looking at the median projection of the temporal lobe in the sagittal section. Yeah, good. Okay, parietal lobe. Who wants to point to the parietal lobe? Yeah, that would be a correct arrow to the parietal lobe. How about on the other one? Yep, parietal lobe. Occipital lobe. Anybody? Yes, occipital lobe there. Occipital lobe there. Good. What's the name? Somebody shout out the name of this structure right here. Cerebellum. Good. So we've talked about the lobes in the cerebellum now. Let me add another name that is not in here because I ran out of space, but you can annotate your slide. This, what is that structure generically? It's a sulcus, and it's got a specific name. It is the parieto occipital sulcus. So its name tells you what it is. It's the sulcus that separates the parietal from the occipital lobes. That one's got a pretty helpful name. Okay, who wants to point to the midbrain? We can't see it here. Point to it again. Right there. Yep, good. That's the midbrain. Let's see what O says. No, it's giving the name of a sulcus. So P and this is the midbrain. P and Q, what is P? Oh, that's labeling over there. This must be capital P. Right. Yeah, so there are structures here that I'm not asking you to memorize. Superior and inferior colliculus. They've got everything has a name. And I mean, yeah, everything has a name. But some of them you have to know, like midbrain. Who wants to point to the pons? Midbrain, pons. The whole thing is the pons. Pons just means bridge. And like, you know, Pontine or, you know, the Pont Neuf in Paris, which means bridge in French. Pontine means bridge-like. Pons is Latin for bridge. And it's a bridge between the cerebellum and the rest of the brain. The pons is where connections from the cerebellum happen. Okay, who wants to, where's the laser? Who's got the laser? Somebody want to point to the medulla oblongata in this picture? That's the midbrain. That's the pons. Yeah, it is around there. Where do we need to go to get to the medulla oblongata? That's the cerebellum. That's more of the cerebellum, but you're getting closer. That's it. Medulla. You knew it was below the, so just, best way to do it is memorize the order. Midbrain, pons, medulla. Or do it the other way. Medulla is below the pons. Pons is in the middle. No, shouldn't use that. Pons is the bridge that makes the connection to the cerebellum. Medulla is below, medulla oblongata is below the pons. Okay? So we have covered midbrain, pons, and medulla oblongata. Okay, we're doing well. Now, I'm not just going to use these. The reason I'm asking you to learn this anatomy is that these have important physiological functions too. And so soon I will be making connections between what does the precentral gyrus do? What does the cerebellum do? What does the thalamus do? But I want you to know where they are before I start using the language of what they do. Okay, so that's why we're doing this. I suppose I could have done this, mixed up. I could have said, okay, let's talk about the precentral gyrus and then talk about what it does, and then go back to anatomy and talk about the cerebellum. But instead, I just want to use the language of anatomy and then flesh that out with the language of physiology. Does anyone know where the thalamus is? In a midline sagittal section of the brain, the thalamus is right there. So this is the midbrain, and this is the thalamus. Pons, medulla. So medulla, pons, midbrain, thalamus in a midline sagittal section. In other sections, it could be more confusing. Hypothalamus. So what is the word hypo? Below. So where do you suppose the hypothalamus is? Below the thalamus. So this area right here would be the hypothalamus. And these are important structures. You'll be hearing about, let me just introduce, the thalamus is going to be a really important relay processing station for sensory input to the brain and for motor output. A really important relay processing center. And the hypothalamus is going to be the master control of the endocrine system. because the hypothalamus is connected to the pituitary. Pituitary. Okay? Which may have gotten lost in this picture, but the hypothalamus is very close to the pituitary. And we're gonna see that by the end of the semester, big time, in the endocrine section. So we know where the thalamus is and the hypothalamus is. Does anyone want to point to the corpus callosum? Anybody? Yes. So the corpus callosum is really this thing here. That's the corpus callosum. There's a very thin membrane between the two hemispheres called the septum pellucidum. But this is the corpus callosum. Does anyone know what the corpus callosum does?
Connects the the left side to the right side.
It is the major pathway that connects the left hemisphere of the brain to the right hemisphere of the brain. White matter. Axons. Why is it white? Myelin. Myelinated axons. Fast information being transferred between the two hemispheres. Corpus callosum is the connection, white matter, myelinated axon connection between the two hemispheres. So this is obviously the corpus callosum has been cut. Okay, corpus callosum, cingulate gyrus. Okay, let me just point this out. So, does anyone know anything about the cingulate? Okay, we can't see it, so it's a gyrus. We cannot see it from the outside, but we can see it on the midline sagittal section, right here. This gyrus, just above the corpus callosum, is the cingulate, spelled this way, gyrus. We're going to see, it's going to be an important emotion control center. We actually cannot see the hippocampus in any of these images, but it's on the slide because we will be talking about it. Any questions about these terms to know, how to know them, what we will be doing with them? Go ahead.
Cingulate gyrus, because we don't see it connecting.
Right.
In the sagittal section.
Because that's
But
right.
does it, like, that? Because we can't see it connected as
So
the
this-
one gyrus in this picture.
So the cingulate gyrus, you're talking about connecting from the left side to the right side?
Yeah, as
It
in
doesn't
like
do
a
that.
one continuum.
It's-
Yeah,
So
okay.
the corpus callosum connects the two hemispheres of the brain, but the cingulate gyrus doesn't. There's a left cingulate gyrus and a right cingulate gyrus.
No, I meant, you know how in the gyrus you can kind of, it's a continuous,
Yeah.
but in the cingulate one, it's fragmented.
Oh, I see. So we have an anterior cingulate and a posterior cingulate. Anterior cingulate gyrus, posterior cingulate gyrus. I'm not going to ask you to know that. Okay, so some of the language of neuroanatomy. It's kind of fun once you get it. Any gyrus or a specific gyrus? Okay, so here is the central sulcus, Q. Let me just double check, Q.
It's D, isn't it?
This is the central sulcus, so this would be the pre-central gyrus. This lump here would be the pre-central gyrus, and this would be the post-central gyrus. Lump between, so here are the two sulci that define the pre-central gyrus.
So whenever they get gyrus, is there gonna be a sulcus between?
Yeah.
The games.
So here is the cingulate gyrus, and this is the cingulate sulcus. Okay, so let's go on and spend the last few minutes of the class doing some coronal neuroanatomy. So this is a section, this is a cartoon from your textbook of a section through the brain. Whoops, I didn't mean to do that. Through a coronal section, right? So it's important to get our orientations. Whenever you're looking at a coronal section, you're looking at somebody else's brain. So this is the left side of the brain, and this is the right side of the brain, right? And so you usually don't have to know left and right in this class, but sometimes it's important to know which one you're looking at. So even though this is on your right, this is the brain's left, because you're looking at this brain, not right. So, in this image, here is the corpus callosum connecting the white matter tracts, connecting the two hemispheres of the brain. And here are the two thalamuses, two, the left and the right thalamus. Below the thalamus, hypothalamus, and below the hypothalamus, the pituitary. Let's introduce another thing that we didn't see in any of the previous slides, the so-called ventricles. Ventricles are fluid-filled empty spaces. They're not empty, they're filled with fluid. They're filled with cerebrospinal fluid. And we can see two lateral ventricles just below this corpus callosum, and a third ventricle here separating it sort of in the thalamus and midbrain area. Cerebrospinal fluid circulates from the ventricles, and then it will actually go into the central canal of the spinal cord. So that thing on the previous slide, this membrane, the septum pellucidum, is shown here. The septum pellucidum separating the two ventricles. So on this side we can see the ventricles, and on this side we can't see the ventricles because the septum pellucidum is obscuring the ventricle. So there's a ventricle behind it. The lateral ventricle is behind this septum pellucidum. One thing I do want you to, so now in the coronal section, we're going to see white matter and gray matter. The gray matter is on the surface, generally, and the white matter is deep. White matter are the myelinated axons, that's what makes them white. Gray matter is where the cell bodies and dendrites of neurons are. That's where the synaptic connections are. So synapses, cell bodies are in the gray matter. Axons, myelinated axons especially, are in the white matter. The gray matter, so this is a cartoon from your textbook that blows up the gray matter, and you can see cell bodies and dendrites, dendritic processes. There are layers of the gray matter based on where the cell bodies are. And you might hear about, in the future, you might hear about cortical layer 4. well, that's gray matter layer. I'm not gonna ask you to memorize which cell bodies are in which layer. There is a layering structure of the gray matter of the neocortex, of the cortex. So on Thursday, we'll start with, what's the name of this section? The coronal section. And we'll start with some coronal sections, and I will ask you, I will pass the slide, the laser around, and you will point to something and give me a name for it. Okay? That will be on Thursday. Remember, the quiz doesn't have any of this. The quiz 1 is on the muscles that you've been learning about in now. Okay, so you've got 2 homework assignments for this class. Do the quiz, and then get ready for class on Thursday. Let's talk some more neuroanatomy. Okay?